Hi everyone, this is me Dr. Hemanta Dhungana, orthopedic surgeon. Today presentation topic will be on orthopedic basic science. In this video, I'll be talking about the bone cells. Four types of cells found within the bone tissue. Each cell type has a unique nature. There are osteocytes, osteoblasts, osteogenic cells, and osteoclasts. Let's talk about the osteogenic cells. Osteogenic cells are the only cell that can divide. Osteogenic cells differentiate and develop into osteoblasts, which are responsible for forming the new bones. And the osteoblasts deposit bone matrix. When the osteoblasts get trapped within the calcified matrix, they become osteocytes. And you gotta know, osteocytes are the most abundant cells within the bone, which accounts for 90% of the cells in the mature skeleton. The cell which is responsible for bone resorption or breakdown of the bone are called osteoclasts. There is a continual balance between the osteoblast generating new bone and the osteoclast breaking down the bone. Okay, let's go one by one. The first one is osteoprogenitor cells, which are also known as osteogenic cells. They are the stem cells inside the bones that play a significant role in the bone repair and growth. Osteoprogenitor cells originate from mesenchymal stem cells. They become osteoblast under low strain and high oxygen tension. They become cartilage under immediate strain and low oxygen tension. They become fibrous tissue under high strain. So the environment will determine their function. Osteoblasts. Osteoblasts are the bone generating cells, B for buildings, you can remember. They are derived from the undifferentiated mesenchymal stem cells. They produce type 1 collagen, alkaline phosphatase, osteocalcin, which is stimulated by 1,2,5-dihydroxyvitamin D. Osteocalcin and alkaline phosphatase are the marker of the bone formations. A RANK ligand. RANK ligand is secreted by osteoblast and binds to a RANK receptor on osteoclast precursor. This is very important. We'll be talking in coming slides. So the osteoblast is upregulated by estrogen, 1,2,5-dihydroxyvitamin D, and it's downregulated by parathyroid hormones, glucocorticoids, prostaglandins, and leptin. So let's talk about the RANKL-RANK-OPG signaling pathway. So to break down the bones, osteoclasts need to be activated. So how does the osteoclast will be activated? When the RANK receptor in the osteoclast precursor bind with the RANKL, then the osteoclast will get activated and the bone resorption or breakdown of the bones takes place. RANKL is expressed by osteoblast, whereas the RANK is the receptor in the osteoclast precursor. RANKL stands for receptor activator for nuclear factor kappa B ligand. However, OPG, which stands for osteoprogerin, it binds with RANKL and it inhibits RANKL activity. So OPG is protective against the bone loss, which slows down the activation of the osteoclast. There are several clinical implications of this, such as osteoporosis, which can result from the loss of functions of OPG gene, and osteopetrosis, which is caused by genetic defect in the absence of osteoclastic bone resorptions. You can also see in the Paget disease, which is caused by alterations in the cytoplasmic binding to RANK or mutations of the OPG gene, or in the osteolytic bone metastasis. RANKL is produced directly by the cancer cells. So the parathyroid hormones stimulates osteoblasts to secrete more RANKL, and when the RANKL binds with the RANK receptor in the osteoclast precursors, the activations of the osteoclast takes place and the more bone get reserved. That is a breakdown of the bones. And the OPZ, it inhibits osteoclast activity. You got to know that. In osteoblast receptors, you mainly have to know PTS receptor and 1,2,5-dihydroxyvitamin D receptors. PTS bind to osteoblast, the cell responsible for creating bones. Binding stimulates osteoblasts to increase their expression of RANKL, and RANKL binds with the RANK receptor of the osteoclast, and osteoclasts get activated and the bone resorption takes place. And 1,2,5-dihydroxyvitamin D receptor stimulates matrix productions of the bone-specific proteins, which is called osteocalcin. What are osteocytes? Osteocytes are the former osteoblasts trapped in the matrix they produced. They account for 90% of the cells in the mature skeleton. They help in maintaining bone and cellular matrix. They are important in regulations of calcium and phosphorus concentration in bone, and it is stimulated by calcitonin and inhibited by parathyroid hormones. You have to know osteocytes do not express alkaline phosphatase. Osteoclasts are the multinucleated, irregular shaped. They help in breaking down the bone. They are the bone reserving cells. They originate from the myeloid hematopoietic cells from monocytes or macrophage cell lineage. And the bone resorption occurs at the ruffle border. And the site of the bone resorption where ruffle border meets bone surface is known as Hossip's lacunae. Osteoclasts synthesize started resistant acid phosphatase, which is an osteoclast enzyme. It breaks down the mineral portion of the bones attached to bone surface via integrins on the osteoclast surface, have a specific receptor for calcitonin, which inhibits osteoclast resorptions, interleukin-1, which is a potent stimulator, which is found to have a role in osteoarthritis. RANKL, expressed by osteoblasts and tumor cells, activate osteoclast, responsible for bone resorption in multiple myeloma and metastatic bone disease. So the osteoclast is stimulated by interleukin-1, RANKL, and inhibited by calcitonin, interleukin-10 biphosphonates.